Hi everyone, it's Dr. LeBlanc with Unit7G, Mutagenesis and Repair, the expansion of trinucleotide repeats and neurodegenerative diseases. Our learning goals are to understand the molecular basis of triplet repeat expansion or TNRE diseases, to understand what polyQ or polyglutamine neurodegenerative diseases are, to understand Huntington's disease as a model for TNRE disease, to understand what genetic anticipation means and its molecular basis, and to understand how the expansion of triplet repeats occurs during DNA replication. Mutations due to triplet repeats are found in several human genetic diseases that are caused by an unusual form of mutation called trinucleotide repeat expansions. Sometimes they're called polyQ diseases because some of them have multiple copies of glutamine or Q. So this is the codon that's repeated over and over and over again. CAG, which encodes glutamine, which is also in the single amino acid codon code, it's called Q. So they're called polyQ diseases often. There are many different types of TNRE disorders. Here's a bunch of them here. We'll talk about them in a little bit more detail in a minute. Huntington's disease is one of them. It has a repeat sequence that I just showed you, CAG. It's in the coding sequence of a gene that can be important. We'll talk about that in a little bit. If you have up to 37 copies of the CAG in a row and the repeat, you can be unaffected, but the number of repeats found in affected individuals is between 27 and 121, so it's a lot of times that this is repeated. It is inherited as an autosomal dominant disease. It is a neurodegenerative and it shows anticipation in males. Most prevalently, usually. So Huntington's disease is characterized by involuntary movements, behavioral changes, and dementia, all that develop gradually. We're not entirely sure how the Huntington protein causes this disease when it has all these extra repeats in it, or what the protein itself functions in normally in the cell when it's a normal protein. But it is found in mitochondria and microtubules and transport vesicles and synaptic vesicles. So it may have something to do with transporting these vesicles around the cell. This is what a brain looks like when from an individual who has Huntington's disease. You can see the neurodegeneration, these large gaps, and the atrophy of the brain tissue. N-terminal fragmentation of abnormal Huntington protein accumulate in the CNS and form inclusions, and that is usually an indication that the neural tissue is dying off. So you can have up to 35 codons and still have a normal functioning protein, but if you have more than those of this glutamine repeat, then the proteins are malformed and have to be dealt with. And the process of dealing with those proteins is what causes the neurodegeneration here. When the length of the trinucleotide repeat increases above a certain critical size, you get your disease symptoms, and the rate of the expansion also increases through generations. That's an unusual characteristic of this gene, is that at every generation, this repeat, once it goes beyond a critical length, just keeps getting bigger and bigger and bigger. So this individual has shown that it has 11 repeats, and here's one with 18, and you can see it just repeats over and over and over and over again. So these are all polyQ diseases with some very long names. Spinobulbar muscular atrophy, spinocerebellar ataxia, all different kinds, mutations. These are different genes that are mutated. these all have polyQ repeats. There are also some trinucleotide repeat expansion diseases that are not polyglutamine, and they have different codons that are repeated. But they're similar in that the longer the repeat, the more severe the disease, and the process of making those repeats only increases the more repeats you have. So here's a graphic showing Huntington's disease. If you have less than or equal to 35, you're going to get different numbers from different sources here. You have a normal allele. If you have greater than 36 repeats, you have the full mutation. Some of these also have what's called pre-mutations. Some of these are in untranslated region, like here for myotonic dystrophy and Fragile X of the gene. This one happens to be in an intron. Huntington's is in a coding region. You can understand a little bit better the mechanism of neurodegeneration from coding region expansions, because they produce proteins that are aberrant and sticky and form these inclusions. But it's not as easy to understand how triplet repeat expansions in introns and in untranslated regions cause the symptoms that they do. There are some very unusual features of trinucleotide repeat expansion disorders. One is anticipation. As the triplet repeat expands, as it gets longer, the severity of the disease tends to worsen in future generations. And the age of onset decreases. So you have a double-edged sword here. You have the disease gets worse in every generation because the triplet repeat is expanding at every generation, and the age of onset decreases. So younger and younger and younger individuals in a family tree end up having or showing symptoms of the disease. This is a similar graphic to what we just saw. If you have greater than 60 repeats, you're going to end up with a juvenile onset. Also, the severity of the disease depends on whether it's inherited from mom or dad. In Huntington's, the TNRE is more likely to occur if it's inherited from dad, and for myotonic dystrophy from mom. So what's happening? How does this repeat expand? Well, there's a lot of good evidence that what's happening during this repeat expansion is that a hairpin is forming after helicase has separated the two strands at the replication fork. This hairpin gets skipped over in one round of replication, and in the next round of replication, it flattens out and gets replicated. So this is what's happening. Here's your TNRE. DNA replication begins and then goes just past the TNRE. Okay, that something happens because of this repeated nucleotides over and over and over again. The DNA polymerase slips off the template and this hairpin forms. Instead of hydrogen bonding to the complementary strands, there's enough hydrogen bonding capability in this between the C's and the G's and the CAG repeat that you can form this hairpin. Then DNA polymerase resumes and DNA and replicates this region that it already replicated. Okay? And then later on, in the subsequent DNA replication cycle, this will flatten out and you will end up expanding this. Sometimes you repair this and the tRNA is the same length, but other times it gets flattened out and expanded. There's a couple of good videos about DNA replication in general, and then DNA replication around these trinucleotide repeat expanding units. So in summary, TNRE diseases have long tracks of triplet nucleotides that have lengthened beyond a minimum range. Huntington's disease is an autosomal dominant neurodegenerative disorder characterized by involuntary movements, behavioral changes, and dementia. Huntington's disease has a CAG repeat in its coding region, and the Huntington protein is found on vesicles in the cell, although its exact function is unknown. Abnormal Huntington protein aggregates to form inclusions in the brain. This may cause the degeneration associated with the disease. Other TNRE diseases have triplet repeats in non-coding regions. How these cause disease is not known. Genetic anticipation occurs in Huntington's disease, which means that the age of onset decreases and the severity of the disease increases as the TNRE gets longer. And the expansion occurs during DNA replication. A hairpin forms in one round that relaxes during the next round, providing additional template for DNA polymerase.